Applications of various staining techniques in research and industry. First of all, introduction to staining techniques. Staining give color in microscopic studies, essential for studying tissue structures used in research and industry. Staining in research. Parameter in almost every field of study. Identifies pathogens and disease conditions, differentiates cells and tissues. Gram staining. Differentiate between gram-positive and gram-negative bacteria, results in different colors, purple for gram-positive, pink for gram-negative. You can see gram-positive gives purple color and gram-negative gives pink color. Acid-fast staining identifies bacteria like Mycobacterium. Endospore staining. Identifying endospores is crucial for identifying certain bacterial pathogens like Bacillus and Clostridium, which cause diseases like anthrax and tetanus. Hematoxylin and eosine, H and E. Most common tissue stain. H and E staining is used in various research areas, including diagnosing cancer, identifying tissue types, assessing structural changes. You can see blood vessels, fat cells, and smooth muscles. Now you can see cancerous cells. Fast staining highlights carbohydrates, glycogens, and mucins, often used to study liver and muscle tissues. Trichrome staining. This stain is commonly used to differentiate between collagen stained blue and muscles stained red. Pap stain. Used mostly in cervical staining, screening. Pap stain allows for early detection and treatment of cervical cancer. Pap stain is also used in the analysis of urine, sputum, and other bodily fluids to detect various conditions. Organelle staining. Mitochondria stained with dyes like Janus green, Mitoview green, and Mitotracker. Endoplasmic reticulum stained with dyes like ER tracker green and red. Golgi apparatus stained with lectins like wheat germ agglutinin. Lysosomes stained with dyes like lysotracker. And now lipid droplets stained with lipid spots, and last plasma membrane stained with lectins. Immunohistochemistry detects antigens in tissue sections, important in cancer diagnosis and immune system studies. Nissl and Golgi staining. Nissl stains cell morphology of cell body of neurons. Golgi stains full neurons for structural analysis. Biomarker staining. Ki67 track proliferation, used for prognosis and therapy selection, used in cancer research. Industry usage. Pharmaceutical quality control. Stains like annexin 5 and tunnel assess cytotoxicity, key in drug screening and development. Food industry. Gram staining detects bacteria in food samples, ensures hygiene and safety in food production. Food industry spoilage detection. Methylene blue and other fungus stains aid in detection. Environmental water quality identifies coliforms, cyanobacteria, protozoa used in pollution and safety monitoring. Now, environmental soil quality. Stains assess microbial diversity in soil, used in agricultural and ecological studies. Medical helps identify pathogens in clinical samples. Right Giemsa stain differentiates blood cells used to detect infections like malaria. Forensics biological evidence. Crystal violet and DAPI staining biological evidence like semen and blood. vital for crime scene analysis. Veterinary, used for bacterial and parasitic pathogen detection in livestock, common in chemical analysis. Future planning. Currently, we have blood stainings, H&E staining facility and camera-fitted microscope. Various students from different departments aware this facility so far. Department of Anatomy and Histology going to start as many staining techniques as possible. All departments will be then welcome to send their samples for staining.